Welcome back. In the last session, we are discussing about the alkylating agents, not only single alkylating agents. This is the same molecule which does double alkylation, and we have found that we have studied or we have seen that it is the guanine mainly which is preferentially alkylated, and that causes double alkylation, that means intramolecular or intramolecular. Final outcome is that the transcription process or the replication process is stopped because the DNA cannot be unwind in that position where it is deformed. Okay. Now, these alkylating agents that we have talked about is either you started with the dihalo compounds, or I showed you towards the end that there could be aziridine compounds to, because aziridines are not that unstable. It will be unstable when it is attached, it is positively charged azidinium ion, or there could be electron withdrawing groups attached to it. Okay. So, I gave you some examples of a quinone and an imine type of a triazine compound, where the aziridine ring is more susceptible to ring opening. Another important drug is cyclophosphamide, which is a prodrug where the nitrogen lone pair is locked with that phosphate, the phosphate group, and then after once that phosphate is released, then the nitrogen lone pair is free, and that can do the alkylation of the DNA. Now, there are other different types of alkylating agents possible, like some are called nitrosoureas. They are the class of compounds belongs to what is called nitrosoureas. Okay. Why urea? Because urea is NCON, urea is NH2 CONH2. So, these are derivatives of urea. So, one is lomustine, another is carmustine. Okay. How do they work? This ureas, if you have read the preparation of the diazomethane, one of the method of preparation of diazomethane is addition of base to N-nitrosomethyleurea. Okay. And so, in this case is also very similar type of reaction happens. If you if it is little bit basic, then what happens? The base abstracts this hydrogen, which is pretty acidic, that bond goes here, this comes here, and that takes the hydrogen. So, that will form this diazohydroxide. Remember, one arm is still the chloro. Now, what will happen? This will generate this diazonium ion by removing the wedge, and ultimately you because these are aliphatic diazonium compounds. So, they lose nitrogen. So, you have a plus charge here. Okay. So, once there is a plus charge, so one of the guanine base can attack this plus. So, it undergoes alkylation. In fact, diazomethane, all diazonium salts are very good alkylating agents, and then you in addition you have this chloride. No? So, now the Y can come and attack the chloride. So, you get a cross linked DNA. So, now the same fate will happen to this DNA, that it cannot do the proper transcription. Okay. So, these are nitrosoureas. I am quickly just covering, trying to cover as many drugs as possible. Then you have these natural products which are called, which is one of the natural product is mitomycin C. Collectively they are called mitomycins. Okay. Now, mitomycins, the structural pattern, I will say that it has got a quinone here, then a 5-membered ring, and then that is a bis 5-membered ring, which is nothing but pyrrolidine, and on the end there is a, this is interesting, there is a aziridinium ring. Okay. Now, what happens if you look at this structure, which is nothing but a quinone, this is a quinone. If you look at the quinone, quinones are electronically they are electron withdrawing. Okay. They are electron withdrawing. Okay. And on the other hand, if you reduce this quinone, and that reduction in the biological system can take place, you have a lot of reducing agents, oxidizing agents, like cyclophosphamide is oxidized, hydroxylated, and then that is a prodrug that is converted to a drug. Here, the first step of action of mitomycin C is it is reduced by a NADH or NADPH mediated enzyme. So, it will become a quinol Now, there is a vast difference between the electronic character of quinol and quinone. Quinone is electron sink, on the other hand quinol is electron source. So, now what will happen, electrons can start flowing in this direction, okay, because now it is electron rich. Before that, what happens, this nitrogen lone pair, as soon as it sees that there is a, earlier the nitrogen lone pair is also feeling the influence of this quinone part as an electron sink. So, the lone pair is kind of locked there. As soon as it becomes quinol, the lone pair is now free. So, the lone pair will now can kick out this methoxy, the bridge that fusion, the methoxy present at this carbon, okay, the ring junction. And then once it forms the plus, so the lone pair now can start migrating from here. Okay. So, basically one step, there is one step which is missing. So, I can write that. So, this is the wedge, that is wedge, and you have this is nitrogen, and then there is another ring here, and there is a aziridine ring. Okay. So, now this nitrogen is plus and there is CH, there is CH2 OCO NH2. Okay. So, what will happen now? As I said, this is plus. So, now the lone pair can, sorry, let us see, nitrogen lone pair. So, this is plus, sorry, before that another hydrogen shift will take place. This hydrogen will come back. I will just erase this. This is yet to be yet to show its electron source character. Okay. So, once this is formed, that is what is shown here, the double bond. You see the double bond is here and the nitrogen is neutral. Okay. So, that step is missing here, one step, there is a step jump here. Okay. So, this is the double bond. Now, what will happen, there is a good electron relay system to open up the aziridine ring, because that goes here, that electron pair comes here, this goes here, and the aziridine ring opens up. Okay. So, when the aziridine ring opens up, what will happen? This will be NH2 and there is a double bond, and this is now a carbonyl. So, it is become an electron sink now. So, the DNA, it could be, I told you it could be, here it is written NH2, but if it is guanine, that will be the ring nitrogen. If it is cytosine, that is also the ring nitrogen. But you just say that DNA, better DNA with a lone pair, that is all, and then that adds here, okay, and it flows back. So, that becomes quinol again. Where is that? So, the ring opening, then the DNA adds. So, you can write here DNA, only DNA, and DNA is plus here. Okay. Then another DNA, this is also become allylic. If you look at that, this situation here, earlier it was not an allylic carbon, but now you have the double bond here. So, that becomes allylic. So, this is also very susceptible, you know, allylic carbons are very susceptible to nucleophilic attack. Okay. It could be by SN prime reaction, that it can attack the double bond and then subsequently that can leave, or it can be just direct SN2 reaction. In this case, it is believed that there is a direct SN2 reaction. So, that another base from the DNA, same DNA, attacks the methylene and this goes off. Okay. So, basically resulting in the formation of this type of molecule. Okay. Well, here they have used the guanine amine nitrogen. Okay. This guanine amine nitrogen, but in many of the other textbooks it will be that this nitrogen is the one which attacks. Okay. However, because it is written in the book, so we have to go by that. This is the book of G. L. Patrick. In that book, this is what is written, but that is not the important issue. Important point is that the DNA is cross linked, whether it is through this guanine nitrogen or this is the amine nitrogen, that is that could be debatable, but the overall effect remains the same. Like this is attached to the DNA and that is also attached to the DNA, and these DNAs are basically present in 2 strands. Okay. So, basically that is a bisalkylation, same effect, but with a different kind of molecule. Okay. Now, why do you need so much architecture in the molecule? Just to give some specificity to the DNA itself and not to other that this molecule does not attack other molecules. Okay. What is mitomycin C? Is it a drug or is it a prodrug? This must be a prodrug, because that needs to be activated by reduction. Okay. And then it shows its alkylating power. Okay. Now, let us come to the chain cutters, okay, or the artificial scissors. Artificial scissors, I told you that they are the ones which will directly cut the DNA into pieces. That is the most direct approach to destroy a cell. Okay. Now, what type of molecules can cut a DNA? There are various types of molecules. One I will show that which is very recent, in the last 20 years or so this has happened, that there are certain molecules which can produce under the biological condition, which can produce what are called parabenzine or benzene 1,4-diradical. Okay. So, what it does? It abstracts, because it is a diradical. So, it will abstract hydrogen from hydrogen donors. Okay. If this diradical is formed right inside the cell and right in front of the DNA, then what will happen? Whatever is surrounding it, it tries to abstract the hydrogen. So, if it is right in front of the DNA, so it will abstract hydrogen from the DNA. Now, this is formed from a system which will be which is called, maybe I can write here, which looks like this. If you hit this system, it will go to the diradical, okay, benzene diradical, and then abstract the hydrogen and form the benzene. The driving force is obviously the formation of the aromatic system. However, this reaction only occurs at room temperature if it is confined as a cyclic system. Okay. And the natural product that was isolated from soil organisms looks like a big structure, but it has that what is called the indienes, that framework is there. Okay. And then it has got this enone part, then a trisulfide, and then this complex carbohydrate system. Okay. So, basically we can say that we can identify 3 important parts. One is indiene, another that forms the diradical, and the trisulfide. Okay. That is a very bizarre kind of molecule, because I do not know any other natural product where trisulfides are present, but this is certainly one. And then this complex carbohydrate. Now, what are the functions of this? The complex carbohydrate that gives binding to the that provides binding to the DNA. Okay. So, that it becomes selective to the only cutting the DNA and not other molecules, that is number 1. Number 2 is this trisulfide. This molecule is stable so long as this remains in this form, but in the biological system what happens, this trisulfide sulfur sulfur bonds are very labile, like the peroxide O bond. So, a biological nucleophile like glutathione, glutathione is a tripeptide where there is a cysteine. So, cysteine containing peptides, that sulfur, cysteine sulfur can attack the middle sulfur and release the other sulfur atom. So, S minus, a thiolate is released by attack of the cysteine sulfur, possibly in the form of glutathione. Glutathione is a tripeptide containing this cysteine. Okay. Then this sulfur minus, because there is this possibility of conjugate addition. So, there it can come back and attacks this double bond. Conjugate addition means Michael addition. So, once there is a change of hybridization, see there is a change of hybridization as you do the, I am not writing the whole structure, it is extremely difficult and very complicated, but I can make it little bit simpler, that you have, what you have is basically a S minus, and this S minus attacks here, and that results in the conversion of the sp2 carbon into an sp3. Okay. And once it forms an sp3 carbon, then this connection between the two terminal acetylenes, where this is what is called a Bergman cyclization, BC, Bergman cyclization. So, that forms a diradical. This will form diradical provided they are sufficiently close enough. Okay. Again I repeat the mechanism is that this molecule is perfectly stable as such. Once it binds to the DNA and once it is, the trisulfide linkage is broken to form a thiolate, the thiolate goes and adds in a like this thiolate, this is what is shown here, thiolate adds in a conjugate fashion, okay, the Michael addition, and so there is this change of hybridization here that brings these two carbons close by, okay, and so that they can react and forms the diradical. So, this is a radical and this is a diradical. This is a radical. So, this diradical species now abstracts hydrogen, because the whole thing is already placed on the DNA via this complex carbohydrate molecule. So, all the things are important here. If you take out the carbohydrate molecule, it will lose the specificity. If you take out the trisulfide, then it will not be a prodrug, it will be very unstable and it will show the Bergman cyclization from the very beginning. Okay. So, it has to be, all the three parts are very important. Okay. Now, this is this is called this natural product, which is here. There are different types of natural products. The natural product that is shown here is called caliciamycin. Okay. Now, caliciamycin is extremely toxic, cytotoxic. Cytotoxic means very powerful, it works in picogram to kill a cell. And then, so you have to somehow make it, although it binds to the DNA, but still its extreme toxicity did not allow it to become an anti-cancer drug, because the question is why it will be specific for cancer, for cancerous cells and not the normal cells. That is true for all the drugs, by the way. That is true for all the anti-cancer drugs, that they can, we are talking about that they are killing by bisulcylation, they are stopping the cancer cell from dividing, from copying, from transcription, from replication. But what will happen to the normal cell? One simple actually there is no shortcut, not much of a shortcut. However, in recent years drugs are made with tailored specificity to the cancerous cell and not the normal cell. But earlier the simple philosophy that one who runs faster, he will reach first the target, he will reach the end point faster. Okay. So, the cancerous cells, because they are rapidly growing, so if you add something which destroys the cells, it is the rapidly growing cells, they will be affected first and the slow growing cells will be affected last. Okay. So, that is on that principle or on that premise the anti-cancer drugs work. Okay. So, if you see that somebody is taking chemotherapy, means with these kind of drugs, usually he or she also loses the hair, because the hair cells grow very fast. So, fast growing cells, but hair fall is not a problem. Once you stop the anti-cancer drug taking, then you can again regain the hair growth. Okay. But to make it really very specific, apart, because that relies on the growth of the cells, the rapidly growing cells will be killed faster than the slowly dividing cells. However, if you give the dose for sufficiently long time, then what will happen? It will ultimately kill the normal cells also, and that is one of the major drawback of chemotherapy, that apart from the cancerous cells, it also hits the normal cells and then kills the normal cells or cause mutation in the normal cells. So, they are potential and cancerous cells later on in future. So, that is major concern about the chemotherapy, but nowadays what happens that the cancer drug like calicheamycin, it is attached by this disulfide bond. I already told you that it becomes a thiolate, and the thiolate triggers the formation of the di radical. This thiolate is attached to a hydrazide linker, CONH2 is a hydrazide. Okay. Disulfide attached to the thiolate is attached by a disulfide to a hydrazide linker, and to the hydrazide linker an antibody which is specific. What are antibodies? Antibodies are basically specific to cells. So, some antibodies you can attach which are very specific for the cancer cell that one person is suffering. So, basically what you do, you take the cancer cell from the person who is suffering and then inject it to, I think, an animal, get the antibody, but that is an animal antibody. You cannot actually inject that into the human. So, you have to, there is a process called humanize the antibody. That means, you have to now do some tinkering on the antibody to make it acceptable by the human body. Okay. So, now if you attach that antibody to the drug, so what will happen? This antibody recognizes the cancer cell and the drug is released by breakage of the disulfide bond. The drug is released right at the in the cancer cell. This is the, so the drug that has been made is called Mylotarg. This is nothing but a monoclonal antibody, that also you know, monoclonal antibody, but which is specific for the cancer cell and which is a human antibody that is attached to the drug. In this case, the drug is calicheamycin. Okay. So, there are many drugs today available which are extremely specific, because these drugs are attached to the antibody that recognizes the cancer cell and delivers the drug only to the cancer cell and not to the normal cell. However, these drugs are extremely expensive because of this monoclonal antibody production and then attachment. So, lot of chemistry and biochemistry involved here. So, you see another important aspect of monoclonal antibody. We discussed it in the context of synthetic biology, catalytic antibody, but here you see monoclonal antibodies are useful for targeting the cancer cells. Okay. The other way to kill the cells, or target the DNA is by what is called the photodynamic therapy. Photodynamic therapy is basically you take a compound which absorbs light, and light not in the UV range, not in the near infrared, far infrared. That means, it absorbs suppose the red light, okay, light of higher wavelength. Okay. Higher wavelength, there is a means profit of using the advantage of using the higher wavelength of light, because if you have a tumor and then the problem is the penetration, that if you shine light to destroy the tumor, this is actually photodynamic therapy, means using light to destroy in therapy, that is photodynamic therapy. In cancer, that can be used. How? That this molecule absorbs light of higher wavelength. Okay, I was just talking about why higher wavelength, why not the lower wavelength, why not green light, why only red light, or it is better if you can cross the red light also, go to the light of even higher wavelength. But advantage is that it has got higher the wavelength, the greater is the penetration power. Penetration power means, because tumors are solid mass. So, if you shine light to activate a drug, so the question is how far this light goes. Okay. So, the higher the wavelength, the greater is the penetration. And this type of molecule, this is a kind of porphyrin, these are called porphyrin molecules, tetrapyrrole in a cyclic framework. What they do, that if you shine light, then it absorbs the light, goes into the excited state, photo excited state, and then transfers the energy to oxygen, which is in the triplet stage. In the triplet state, it is very unreactive, and then it goes to the singlet. So, basically it is an energy transfer. This porphyrin goes to the higher excited state, transfer comes back, releases energy which excites the oxygen. So, oxygen becomes the singlet oxygen, and singlet oxygen is very dangerous for the cell. So, it now destroys whatever cell components are there. So, basically that is one of the example of a photodynamic therapy. But remember, one important point here is, first of all you have to use a drug which absorbs light and generates singlet oxygen, and the singlet oxygen generation can be done by many molecules, but this is a special one because this absorbs at higher wavelength. Higher wavelength means greater penetration, so that the whole, because you have to kill the whole tumor. Remember, in cancer, if ultimately after chemotherapy, if only a single cell is present, cancer cell is present, that can again cause cancer. So, you have to get rid of all cancer cells in the body, because your immunity is not working. So, that one cell cannot be taken up by the taken care by the immunity. So, you have to destroy all the cells. Okay. So, what we have not touched many other targets because of the shortage of time, but I will just point out some of the other targets, which one is called matrix metalloproteinases. These are zinc dependent enzymes which degrade the extracellular matrix and encourage the process of angiogenesis, tumor progression and metastasis. Matrix metalloproteinases, they are important for what? They encourage this process of angiogenesis, tumor propagation. What is angiogenesis? That means, if you have the cancer cell like this, so when it grows, it has to grow the blood vessels in different directions, and then because the cancer needs more nutrients than the normal cell, because it has to grow very rapidly. These nutrients, to get the more nutrients than the normal cells, it has to grow a number of blood vessels, capillaries, and this is a process what is called angiogenesis, and angiogenesis is supported by this matrix metalloproteinases. So, you can, if you can develop inhibitors of matrix metalloproteinases, that can be good anti-cancer agent. In fact, some of the non-peptide inhibitors of matrix metalloproteinases are available as anti-cancer agent. I think I will also talk about this histone acetylase and deacetylase enzyme. What are these? In fact, this is one aspect which we have not told you in the biochemistry class. The first section is that the DNA, the length of the DNA is quite DNA has a it's a very long piece, but the cell dimension is much less. So, how the DNA is packaged in the cell? The packaging is done by like how our threads are put on the ribbons. So, basically there are ribbons, and the ribbons are made up of the DNA is there, is going like threads which are packed on a ribbon. So, and inside there is this protein. What makes the ribbon? That is called proteins which are called histone. So, histones are nothing but kind of a ribbon like shape, and on this, you know, the solenoid, also you know solenoid. So, you can pack actually a lengthy piece, you can go by with terms, you can package the long DNA into the cell around a protein ribbon or a spindle. Okay. Now, the question is, once it is stuck to the protein, one it is bound to the protein, why it will be binding to the protein, that is one question. Okay. Because DNA is, what is the charge of the DNA? The charge of the DNA is negative. Okay. So, the histone proteins, that means the proteins which makes this histone, the histone proteins must be positively charged. Now, what are positively charged? Arginine and lysine at the biological pH, because they will be NH3 plus, or arginine will be also plus charge. So, the histone proteins have lot of lysines, that what people have found. Lot of lysines makes sense, because the lysines will be NH3 plus, and that makes a very good force between the histone protein versus the DNA. Okay. But at the time of replication or transcription, what will happen? You have to now take it from that you have to detach the DNA from the histone protein. Okay. How do you do that? What you do, the enzyme does acetylation of the lysine. If you do acetylation of the lysine, it loses the positive charge. So, that means the DNA becomes loose now, it can come out, it can detach for the time being, so that your replication process takes place, replication and transcription takes place. Okay. And once it is done, then there is another enzyme which brings it back to the amine form. So, this is what is called histone acetylase and histone deacetylase, both are required. Histone acetylase basically detaches the DNA, allow the DNA to detach from the histone protein, and histone deacetylase makes it again attached to the histone protein. Okay. This is continuously going, acetylation, deacetylation. So, this is directly related to the cell growth, cell division. Okay. So, then that means, if you can find ways to disturb these enzymes by having inhibitors, then you can get anti-cancer drugs. And in fact, today in the market, now these histone deacetylation enzymes, they have a zinc in their active site, they are zinc dependent enzymes. Okay. It is a hydrolysis, deacetylation means hydrolysis, it is a zinc mediated hydrolysis, and so lot of molecules have been made. One I will just tell you, such a simple molecule. This is nothing but what is called 1, 2, 3, 4, 5, 6, 7, 8. This is suberik acid. So, this is a suberik acid based hydroxamic acid. On the other side, there is hydroxamic acid. So, suberenyl, this is aniline. Okay. So, suberic acid, this is aniline, and that is your hydroxamic acid. If you abbreviate that, that is called SAHA. SAHA is one of the latest anti-cancer drug which works on the deacetylase enzyme, and abbreviated as HDAC. HDAC is histone deacetylase. So, SAHA, and then another compound, but this CONH is what actually chelates to the zinc. So, you have to chelate the zinc, but surprisingly these are so simple molecules which are approved in the market, and this is they work by histone deacetylase, inhibition of the histone deacetylase. Okay. So, I think that is we will quickly go through some of the other very important, I think we should, as organic chemists, we should know these compounds. One is called cisplatin. Cisplatin is an anti-cancer, very good anti-cancer drug. I told you about bismetallation at that time, when I was talking about bisalkylation. Bismetallation means that you have a metal ion, you have this DNA, and you have a metal ion where the two bases have coordinated to the metals. Okay. So, this is nothing but a kind of bisalkylation, but the alkylating agent is a metal. So, metallation. Cisplatin is this compound, cis because these chlorines are cis here, Transplatin does not work, it is only the cisplatin. Cisplatin crosses the cell membrane, and then these chlorines are very labile. So, one chlorine is replaced by the water, followed by another chlorine. So, this is the actual active drug. So, this is nothing but a prodrug again. What is a prodrug? Which has to be activated. So, it goes inside the cell, activated into this form. Now, this wedge, the hydroxy ones, the ligands are replaced by the ligands of the two guanines here. Okay. So, that causes this is they showed intramolecular, this could be intermolecular also, like this. This is intermolecular and this is intramolecular. They could be there, if there are two G's one after another, then that could be the structure. If there are one G here and then followed by a different base, another adenine, that also can participate. Remember, guanine is the preferential side, but that does not rule out adenine or cytosine to chelate to the metal ion. So, that is the working principle of cisplatin. Now, let us before finishing, we will talk 2 or 3 of the other important anti-cancer drugs which are recently developed. One is called temozolomide. Temozolomide works on the principle of generation of diazomethane, which is a very strong alkylating agent, that we know. Okay. So, it alkylates the DNA, but it has to be produced inside the cancer cell, that is important. Now, cancer cell has a pH which is much less than the normal cells. The reason for that is the production of lactic acid, lot of lactic acid, and the reason for production of lactic acid is that cancer cell wants lot of energy to grow. So, cancer cells are more acidic, that is the difference with the normal cells, and that is what is exploited by this molecule. This is temozolomide. This nitrogen, so long as it is in the carbonyl, this nitrogen cannot enter into resonance with the azide, okay, N double bond N. Similarly, this nitrogen cannot enter into resonance with this imidazole nitrogen because of this carbonyl. But once you have a protonation here, because the cancer cells are acidic, so it will be preferentially protonated in the cancer cell than the normal cell. So, what will happen? As a result, water will come here, attacks, and this becomes a species like this. And what will happen to the carboxy? Because the carboxy will now be a beta keto type system, beta imino system. So, this is what is the carboxy, and this is CONH2, this will be NH, then N double bond N and methyl. Okay. So, now the carboxy will decarboxylate, like what happens in beta keto ester, beta keto acid, and then what, so you get this species. After once you have this species, now this lone pair comes here, because comes here, and then this is released. This N minus is in conjugation with this double bond. There is a amide here also, remember. So, that goes there, that goes here, then the amide carbonyl becomes O minus. That is the driving force for release of this methyl diazonium salt, and then that kills the cells, okay, by alkylation. Then let us see what is in store for us. Okay. Then there is another very interesting development. In the body, I told you that there are in cancer cells, one difference is the pH. The other differences is that many of the receptors work by attachment of neurotransmitters or by hormones. Hormones go through the whole body. Okay. So, there are receptors that can be overexpressed if you need higher growth. Okay. So, you need to express the receptors for the growth factors more, and because the cancer cells are growing more rapidly. So, many of these receptors which are receptors for growth factors, they are overexpressed in cancer cells. One of the receptor is estrogen receptor, another is androgen receptor. What is this? androgen and estrogen. Androgen is the male hormone mainly, and the estrogen is the female hormone. So, that means many of these cancers which are related to mainly to women, like the breast cancer, that is you can exploit in those cases because their estrogen receptors are overexpressed. So, you have to now develop what are called antagonists of estrogen receptors. Similarly, for male population, androgen receptors are overexpressed, which can be a causative factor for prostate cancer. Okay. So, you have to utilize what are called anti-androgens or anti-androgens. Okay. So, here you have to use anti-estrogen. Now, the question is what is the process? It is one thing that you can develop your anti-estrogen to stop the estrogen receptor, to stop the estrogen from binding to the estrogen receptor. Okay. That is one approach. The other approach is better check what is the biosynthesis of estrogen, and then if estrogen is not there, then how the receptor will be activated. So, there are two ways, either you add an antagonist to the estrogen receptor or you stop the biosynthesis of estrogen. Okay. I think that is clear. So, what we are talking here is the biosynthesis of estrogen. Quite quick, this is what is one androgen. Androgens are basically, this is a steroid molecule. Steroids are typically have 3 rings. These are parhydrophenanthrene rings, and this is a 5-membered ring. That is the typical skeleton of a steroid. Okay. This is what are called androgens, and androgens go to estrogen. What is the structure of estrogen? You will be this is what is the structure of estrogen. So, what happens in this reaction? The methyl is gone and the ring has become aromatic. So, this is aromatic and this is aromatic, and the methyl is also gone, loss of methyl. And they found that the methyl is actually coming out as a formate. So, that means the methyl has been oxidized first to aldehyde, first to alcohol, then aldehyde, and then followed by formate. Okay. So, they started looking at the mechanism of this compound, mechanism of how these androgens are converted into the estrogen. Okay. And finally, they found out that it is the enzyme which is called aromatase, because what you are doing is aromatize the A ring. This is, by the way, the A ring of steroid. This is B, this is C, and this is D. Okay. A ring you have to aromatize. So, the enzyme is called aromatase. It needs oxygen and it needs a NADPH. Okay. So, that is actually the electron donor agents, okay, and itself becomes oxidized to NADP plus. First it goes to CH2OH and then it goes to CHO. Okay. So, step by step oxidation, and then, but it is an iron dependent oxidation. Remember, we have done this penicillin biosynthesis, where this type of iron oxo species are formed, which are very good oxidizing agents. Very similar type of chemistry happens here, and then based on this we can actually now aromatase inhibitors are available in the market. And I remember that when I was doing my postdoc at Hopkins, the work was going on aromatase at the Johns Hopkins Medical Institution. So, one fellow, he was working on this aromatase. At that time, it was only at the academic interest, means molecules, people are writing different molecules, that this can be an inhibitor, that can be an inhibitor. But finally, I am so glad that today you have different aromatase inhibitors in the market. We will discuss this aromatase in the next session, because still there are some chemistry, very interesting chemistry that are associated with this aromatase. Thank you.